Hey, if you didn't already figure out how to use this thing, let's go over it real quick. This is a codon chart, very similar to the one you guys already are pretty good at using. Okay, this one's easier, I think. Okay, and this is the one your book has, and this is the one that's on those worksheets I just gave you. So I wanted to show you what it does. Again, what this whole thing is, you take a codon, which is on the RNA, 3 letters, and you use this chart to figure out what amino acid those 3 letters code for. They use CAC as an example. Here's the rule of thumb with this thing. You start in the middle. If it's CAC, like I said, if this is the mRNA codon right here, if this is the codon, CAC, you start in the middle, you find the C, A is next, you stay in the C area, you work your way to the A, and you find the C, histidine. So CAC codes for histidine. See if you guys can figure out the next one. UCC. I know it's kind of hard to read.
Sure.
It's small. U is the first letter, start in the middle. Then you go to the C, then you go to the C, and that word says serine right there. Good. Okay. Last one, what's it got? AAG? AAG, lysine. I think this one's easier to read because you don't have to kind of sit there and go here, here, and here and figure out where they all meet up. Yeah, this one's actually easier to read, but I noticed that when it comes to most biology books, they like to use that other one. I think this one's easier. I think your book got it right. So, but I wanted to be sure you know how to kind of use both of them. All right, that's one. Let's play another kind of game here. Kind of treat today like a game. Now, you guys were doing that worksheet just now, and you kept asking me, what does it mean by type of mutation? That's what I'm going to go over here right now real quick. It's easy. Let's say I have an original piece of DNA. This DNA is correct. Okay, this is right. I want you to see how this is different from the one above. You guys look closely about. Basically, I'm just asking you, how is this bottom strand different from the top strand?
I have to have them separate.
Is there a stop right in the middle?
Don't worry about stop. I'm just looking at sheer letters. What are the letters? Don't even get, I mean, literally.
There's a G paired up with a T.
That's right. Instead of a G, there's a T there. That's a mutation. This type of mutation is called a substitution. Think about it. You get a substitute teacher. You get one teacher substituting for another teacher. It makes sense. This is called a substitution. You have one letter substituting for another letter. So sometimes this happens in your DNA. Let's go to the next one. Compare this one and this one. So ignore the one in the middle. Take this one, and how is this third one different from the first one? There's an extra G there that shouldn't be there. You guys see that? It wasn't up on the top one. There's an extra G. This is called an insertion. You insert something that shouldn't be there. Okay, it's called an insertion. All right, one more to go.
There's no A.
There's no A. Very good. There should be an A right there. This is called a deletion. It's like hitting the delete button. Yeah. Okay. So basically on that sheet of paper that you guys have for homework, when it said type of mutation, you'll write down one of these blue words depending on how that DNA sequence is different from the one at the top of the page. So it's either going to be a substitution, an insertion, or a deletion. Okay, it's pretty easy to figure out. I mean, you just kind of have to eyeball it and see how they're different, but go ahead and jot that down real quick. Again, this chapter's kind of fun as far as science fun can be.
So why?
All you're doing on this whole point of this activity was to look for mutations. I should kind of put the word at the top, mutations. You're looking for what's different from these strands versus your original strand. How is it different? And this is kind of showing you that every once in a while, your DNA can get messed up, and these are the type of mess-ups that can happen.
Oh, so they're all in the same spot.
Yeah, they're all, no, not necessarily. They're all kind of in different spots, but there are different things happening. One of them, this one has one letter being substituted for another. This one you're actually putting in an extra letter that shouldn't be there. This is one you're taking out an extra letter. These all vary from your original sequence.
Oh, that's probably not extra.
Yeah. All right, I'm going to kind of move it up here. Types of mutations fall into two major categories. One's called a point mutation. Up above, when we were talking about substitutions, that's a point mutation. That's considered to be a point mutation because you're changing one base pair. You've got one thing wrong and something else is in its place. It's a point mutation. But look at, by having a point mutation, I'm going to group these in groups of 3. By having a substitution, point mutation, you're not messing up the reading frame. You're not messing up, like look at, even though there's a mutation right here, yes, you're going to mess up that amino acid, but you're not going to mess up the amino acids after it. That's what frame shift mutations are. Frame shift mutations can be kind of bad. Insertions and deletions are examples. Deletions are examples of frame shift mutations because they shift the reading frame. And if you have no idea what I mean by when I say shifting the reading frame, let's look here first. Look what's happened in this next sequence. By adding an extra G, you have now changed every codon after that. That's not going to code for the right amino acid. That's not going to code for the right amino acid. In other words, you've like picked it up and like shifted everything over. If you don't understand that, wait, where's that one sheet? I think your book actually uses this as an example. Hold on. Can I drop a sheet here? Yeah. Check out this sentence. Oh, sorry. This is what I mean by a frame shift mutation. Look at the top sentence. The big dog ate the cat. Okay. Look at what I did on the next line. All I did was insert an extra I. When I put in an extra I, all the other letters shifted over, and now it just reads gobbledygook. That same kind of thing happens in DNA if you add an extra base that shouldn't be there. It's going to shift everything over, and every amino acid after the mutation's going to get screwed up because it screws up your sets of 3. Does everybody kind of get that? Yeah, so even though the same letters are there, look at D-O-G, A-T-E, T-H-E, C-A-T, it won't read the same. It won't read the same in DNA. And that's why when you have an insertion or a deletion, that's what I mean by it's going to mess up the reading frame. Those are not going to code for the right amino acids anytime after the mutation. You kind of can get lucky with a substitution. Sometimes you can have a substitution, and if you're really lucky, you'll maybe even possibly code for the same amino acid. But certainly, your codons afterwards won't get messed up. So, yep, those are the 3 types that will help you on that one worksheet to figure out which ones those are. And finally, one more sheet, guys. This is pretty easy stuff, too. This is it, as far as the notes. We're going to talk about mutations, since that's the word of the day. When somebody says there's a mutation, even in Hollywood, in all the movies you see, when they talk about, well, there was a mutation in that, and the person ended up, like Spider-Man or whatever. Okay? A mutation is when your DNA gets changed, gets messed up. And remember, it has to do with those base pairs, those A, T, C's, and G's. A change in DNA, and otherwise known as a change in those A, T, C's, and G's, is what a mutation is. What causes a mutation? Something called mutagens. And there's a bunch of them. Mutagens are chemical or physical agents that cause a mutation. And I listed a couple there. We talked about these a little yesterday. UV rays, tobacco, X-rays, that kind of stuff. Okay, so those are things that over time will cause little changes in your DNA, and it really only becomes a problem after many of those mutations kind of accumulate in your DNA. Speaking of which, oops. Not all mutations are bad. Some are bad, most are bad. I have a little bad face there. Obviously, most of the time, mutations are going to result in disorders. We've talked about a couple: cancer, sickle cell, Tay-Sachs, all disorders we've talked about. All of those are caused by when one or more letters in your DNA.
Is that African mosquitoes?
Yeah, I'll explain in a minute. When one or more bad things happen in your DNA to change it. Remember, if your DNA gets changed, ultimately what happens, guys, is you're not going to make the right protein. Proteins keep you alive. Proteins form who you are. However, believe it or not, mutations can actually be good. And I have African mosquitoes there as an example. Obviously, these are mosquitoes in Africa, okay? Obviously, mosquitoes, as we all know, are bad. They carry a lot of diseases. So obviously, you're going to want to use a lot of pesticides to get rid of them, and that's exactly what they do. However, there's this group of African mosquitoes. They applied a bunch of pesticides on these mosquitoes, and most of them died, but not all of them. Some of those mosquitoes, turns out, have a mutation in their DNA, which causes those mosquitoes to produce a protein that the other ones don't. That protein somehow interferes with the use of pesticides and keeps them healthy, keeps them alive. So they're resistant. They're resistant to pesticides. Now, obviously, this is good for the mosquitoes. It's not necessarily good for humans, but it's good for the mosquitoes that they have this mutation. So it's good for the survival of them. So these kind of mutations, these good mutations, happen in nature all the time. All the time. There's many kinds of evidence to support that. I always bring up Magic Johnson here because Magic Johnson, as you guys all know, has HIV. They actually believe that Magic Johnson has some mutation in his DNA, which is making him make some protein, which is keeping him healthier than some other people who have HIV. So obviously, I think pharmaceutical companies are looking to bottle up whatever it is he and other people like him are making to hopefully have some kind of cure for HIV. So that's kind of what I've heard is going on now. Hey, there's another word here I'll end on, polyploidy. It kind of sounds like a big word. It's another example of mutations that are good. There are a lot of organisms out there that are polyploidy. If you guys remember, we are diploid organisms. Diploid, D-I-P-L-O-I-D. We're diploid. We have 2 sets of chromosomes in every one of our cells. One from mom, one from dad. Some other organisms don't work that way. They actually have more than 2 sets of chromosomes. Strawberries. This is why we used strawberries about a week or so ago to isolate their DNA. They actually have 8 sets of chromosomes. They have a ton of genetic material. That's just the way they survive. It's a mutation. It comes about during the meiosis process. Their chromosomes don't separate right. Too many get sent one way. But for them, it's key to their survival. That's how they operate. To be honest, 80% of the plants out there are polyploidy. Wheat, rye, watermelons, strawberries are mostly polyploidy organisms. So that's necessarily that's kind of considered an example of mutation where it's kind of good. So that's all, guys. That's all I have for today. And questions, comments, concerns? Keep working on those worksheets I handed out today. This should help you do those activities on there. All right.